Bismillahirrahmanirrahim. Elhamdülillahi Rabbil alemin. Ve salatu ala nebiyhi Muhammed. Research article, Effect of Antioxidant Water on the Bioactivities of Cells. Bioaktiviteler, hücrelerin bioaktiviteleri üzerinde antioksidan suyun etkisi. It has been reported that water at the interface of a hydrophilic thin film forms an exclusion zone, which has a higher density than ordinary water. A similar phenomenon was observed for a hydrated hydraulic ceramic powder, and water turns into a three-dimensional cell-like structure composed of high-density water and low-density water. This structured water appears to have a stimulative effect on plant growth. This report outlines our study of antioxidant properties of this structured water and its effect on cell bioactivities. Yani burada bahsedilen belirli yapıdaki su, farklı bir yapıdaki su, standart sudan hücrelerdeki bioaktivite yapılarında, bioaktivite faaliyetlerinde olumlu etkisi olduğu gözlemlenmiş. Culturing media which were prepared utilizing this antioxidant structured water promoted the viability of raw 264.7 macrophage cells by up to 3 times. Yani bu antioksidant yapıdaki suyun hücrelerin viyabilitesini artırdığı, neredeyse 3 katına artırdığı gözlenmiş. Yani hücrelerin canlılık faaliyetlerini. The same tendency was observed for other cells including IEC/6, C2C12 and 3T3L1. Also, the cytokine expression of the splenocytes taken from a mouse spleen increased in the same manner. The water also appears to suppress the viability of cancer cells. Yani işin güzel tarafı fıtri canlıların canlılığını artırırken kanserli hücrelerin canlılığını da baskıladığını, zayıflattığını gözlemliyoruz. MCF7 tipteki kanser hücrelerine. These results strongly suggest that the structured water helps the activities of normal cells. Yani bu sonuçlar şunu gösteriyor ki bu yapılandırılmış su normal hücreleri kuvvetlendiriyor while suppressing those of malignant cells, yani anormal hastalıklı hücreleri de baskılıyor. Introduction. There have been many arguments on the role of water in a cell and this debate still continues today. Hücrelerdeki suyun rolü nedir tartışması epeydir yapılıyor ve halen de devam etmekte. Most of these debates are still primitive, focused on whether water is a simple solvent medium of these diverse organic components in cytoplasm or it plays an essential role. Bu tartışmalardan bir kısmı hala çok başlangıç durumunda. Özellikle su basit bir etken midir? Sadece bir çözücü müdür? Yoksa çok essential, yani çok esas, lüzumlu, asli bir görev mi almaktadır canlılıkta? As the name suggests, most studies in molecular biology have been conducted focusing on the behavior of molecular components such as lipids, proteins, and other cellular molecules, including various antioxidants. Moleküler biyoloji alanındaki çalışmaların birçoğu adından da anlaşıldığı gibi lipidler, proteinler ya da çeşitli antioksidanların etkileri alanında yapılmaktadır. Despite the fact that water occupies the largest portion of a cell, it does not receive proper attention from academia in biology and medicine, likely because the structure of water and its effect on biological activities is still not well understood. Gerçi canlıların çok büyük bir kısmı su olmasına karşılık su hakkındaki akademik çalışmalar oldukça azdır. Bunun sebebi de suyun canlılık üzerindeki etkisinin tam anlaşılmamış olmasındandır. Recently it was claimed that there is a fourth phase of water similar to a liquid crystal in addition to the three phases of water, solid, liquid and gas. Yakın zamanda ifade edildiği gibi suyun katı, sıvı ve gaz halinden başka dördüncü bir formu olduğu belirlenmiştir. This fourth phase take place at the interface of a hydrophilic material such as Nafion film, as water molecules are arranged along the polarity of the surface. Bu dördüncü faz hidrofilik materyallerin, mesela nafyon gibi yüzeyde artı eksi yüküne ya da polarity, yani kutuplu bir şekilde dizilmesinden oluşmaktadır. Since its density is higher than ordinary water, bu dördüncü fazın öz kütlesi normal sudan ağır olduğu için microspheres in a suspension are excluded as the water is structured, and based on the phenomenon it was named as an exclusion zone. Bu suyun yapısından dolayı, öz kütlesinin ağırlığından dolayı mikro küreler bu yapıdan dışlanır. İşte bu olayda exclusion zone, yani dışlanma bölgesi olarak adlandırılır. Also, electric potential as high as 200 millivolts has been observed to develop across the boundary of the exclusion zone and outside of this region. Hatta bu exclusion zone bölgesinin dış tarafında eksi 200 milivolt civarında da voltaj gözlemlenmiştir. This potential is generated by the dissociation of water molecules into negative ions and protons as it is structured. Bu potansiyelde, yani bu voltajda su moleküllerinin ayrışmasından, yani OH eksi ve protonlar olarak ayrışmasından ortaya çıkmaktadır. Tabii muhtemelen bir tarafta OH varsa diğer tarafta da H kalıyor, yani hidrojen kalıyor. Hidrojen atomu. This important finding implies that water itself can affect the growth and bioactivity of live beings. Bu önemli bulgu suyun canlılar üzerinde büyümeye etki edeceğini göstermektedir. Actually, it was reported that an exclusion zone is formed also around hydrophilic ceramic powder and can stimulate seed germination and early sapling growth in brown chickpea seeds. Aslında hidrofilik seramik tozunun etrafında da böyle bir dışlanma bölgesinin oluştuğu ve bu da brown chickpea seeds, yani bir çeşit bitki, kahverengi nohut tohumlarının stimüle edilmesine, yani hareketliliğine, canlanmasına vesile olduğu tespit edilmiştir. Root length and or formation of shoots increased at least 2 to 3 folds. Yani kök uzunluğu ve sürgünlerin oluşmasında en az 2-3 kat artırma etkisi olduğu gözlemlenmiş. In due course, structured water may also influence cell bioactivities. Yani buna binaen yapılandırılmış suyun hücrelerin bioaktivitesi üzerinde de etkisi olacaktır. Olabilir, may also. To confirm this possibility, an evaluation of the property of structured water composed of an exclusion zone and its effect on bioactivities of cells are necessary. Bunu tespit etmek için, onaylamak için ne yapabiliriz? Bu mümkünatı, yani bu mümkündür denildi. Böyle bir exclusion zone, dışlanma bölgesi ve onun hücrelerin canlılığı üzerindeki etkisinin araştırılması gerekmekte. In this study, structured water formed by mixing with a hydrophilic ceramic powder was discovered to have an antioxidant property. Bu çalışmada hidrofilik seramik tozu ile karıştırılarak oluşturulmuş, yapılandırılmış bir suyun antioksidan özelliği olduğu tespit edilmiştir. In addition to address any arguments regarding the effects of any possible dissolved mineral components, we have developed another method utilizing far infrared ray waves from the ceramic powder to make structured water in a non-contact manner. Bir de herhangi bir birleşim, yani bir suda mineral çözmenin ötesinde suya temas etmeden sadece kızıl ötesi ışınlar göndererek seramik powder, yani seramik tozundan ışınlar göndererek su yapılandırma yöntemi denenmiştir. Bu da non-contact, yani suya temas etmeden. This structured water was utilized to prepare culture media for evaluating its effect on the bioactivities of cells. And this water appears to have a meaningful effect on the bioactivities of normal cells, such as cell viability, phagocytic activity and natural killer cell activity. Yani bu yapılandırılmış su hücrelerin bioaktivitesi üzerinde yapılacak testi gözlemlemek için, ölçümlemek için, bir medya oluşturmak için kullanıldı. Bir ortam oluşturmak için kullanıldı. And this water appears to have a meaningful effect. Yani bu suda, yapılandırılmış suyunda anlamlı etkisi olduğu gözlemlendi. Ne üzerinde anlamlı etkisi var? Normal hücrelerin bioaktiviteleri, mesela hücrelerin canlılığı, fagositik aktiviteleri ya da natural killer cell activity, yani doğal öldürücü hücre aktiviteleri. Conversely, the viability of cancer cell was suppressed ve tam tersi olarak da kanserli hücrelerin canlılığının baskılandığı when it was incubated with the same medium made of structured water, yani bu yapılandırılmış yapılmış suyun ortamına konulduğunda kanserli hücrelerin de baskılandığı, zayıflatıldığı gözlemlendi canlılığının. These results imply that the role of water should be understood as a critical component rather than merely as a simple media in a cell. Dolayısıyla hücredeki basit bir ortam olarak düşünmek değil, hücrenin canlılığını doğrudan etkileyen önemli bir etken olduğu suyun ne yapılması lazım? Imply, yani ifade ediliyor, anlaşılıyor. Materials, ikinci başlık, Materials and Methods, Evaluation of Antioxidant Property of Water. Suyun antioksidan özelliğinin ölçülmesi. 2.1.1 Water mixed with the ceramic powder. Seramik tozuyla karıştırılmış su. A hydrophilic ceramic powder, CeLB, hidrofilik bir seramik toz olan CeLB, with particle size ranging from 40 nanometer to 1 micrometer was obtained from Quantum Energy Corporation Limited. It was produced by finely grinding natural clay minerals of the feldspar family with silica as the main component, more than 60%. Şimdi suyla karıştırılmış bir seramik tozu suyla karıştırılıyor. Hidrofilik bir seramik tozu var. CeLB, muhtemelen bir marka. Bunun da partiküllerinin, parçacıklarının boyutu 40 nanometre ile 1 mikrometre arasında. Quantum Energy firması tarafından sağlanmış. It was produced by finely grinding natural clay minerals of şimdi doğal kil minerallerinin parçalanmasından elde edilmiş bu seramik tozu. Feldspar ailesinin minerallerinden ve with silica as the main component. Asıl main component olarak, ana component olarak da silika mevcut. Yani %60'dan fazlası. To evaluate the antioxidant property, yani ne kadar antioksidan olduğunun ölçülmesi, an ultra weak photon emission was measured. Ultra zayıf foton emisyonu ölçüldü using a photomultiplier. Photomultiplier tube. Hamamatsu Photonics. Under total darkness, tamamen karanlık altında ne kadar foton emisyonu olduğu ölçüldü. Figure 1 shows the schematic diagram of the photon emission measurement system. Figür 1'de foton emisyonun ölçüm sisteminin şeması verilmiş. Figür 1'e bakıyoruz. Komple karanlık bir ortam var. Bilgisayar, sensör, karanlık bir oda. Şurada muhtemelen seramik tozu var. Ve bu karanlıkta bu Hamamatsu tekniğiyle foton emisyonu ölçülüyor. Hamamatsu Photonics ya da Hamamatsu muhtemelen burada bir firma da olabilir. Firma ismi ya da ürün ismi firma. For the evaluation of the antioxidant property of water mixed with the powder, 3 kinds of samples were prepared. Bunu ölçmek için de yani tozla, seramik tozuyla karıştırılmış suyun antioksidan özelliğinin ölçülmesi için 3 tane örnek hazırlandı. The same amount of bean sprout stems were mixed with the deionized water only. Deionized water mixed with natural silica powder and deionized water mixed with this Kelby powder respectively. Yani 3 farklı karışım hazırlandı. Bir tanesi fasulyenin filiz sapları var. Sadece deiyonize su var. Diğerinde aynı miktarda ama bu sefer deiyonize su doğal silika tozuyla karıştırılmış. Bir diğerinde ise, üçüncüde ise deiyonize su Kelby tozuyla, yani bu firmaya ait olan bir tozla karıştırılarak 3 farklı ortam hazırlandı. The concentration of the powder in each mixture was 0.01 grams per milliliter. Konsantrasyondaki toz miktarı hepsinde de 0.01 gram bölü mililitre. The number of photon emissions per 1 second was measured for 8 minutes. And an average value was calculated. 8 dakika boyunca foton emisyonu ölçüldü ve ortalama değer hesaplandı. The measurement was repeated every day for 7 days. 7 gün boyunca her gün bu ölçüm tekrarlandı. In addition, the color change of the bean sprout stems was observed simultaneously. Buna ilaveten de o fasulye filiz saplarının renk değişimleri de gözlemlendi. 2.1.2 Water treated in the non-contact methods. Yani kontakt olmadan kontaksız metotta suyun değişimi. For the non-contact treatment of water, vials containing 50 ml of the deionized water were put up into the ceramic powder halfway for 48 hours before it was used for the preparation of the culturing media. Deionized water was obtained by a water purification system that produced an ultra-pure water type 1 grade with electrical resistivity of 18.2 mega-ohm centimeter and reduction of inorganics up to 99.99%. Young Lin Instrument Aqua Max Ultra 370 series, yani saf su oluşturan bir cihazla, saf su üreten bir cihazla %99.99 oranında saf bir su üretildi. ve bu suyun içine seramik tozu 48 saat. Figure 2 shows the schematic diagram of water treatment in a non-contact manner. Figür 2'de kontaksız nasıl üretildi o gösterilmektedir diyor. Figür 2. Conical tube containing deionized water, container filled with the ceramic powder. Preparation of water in the non-contact method. Deionized water in a 50 milliliter vial was put up into a ceramic powder for 2 days. And used for the preparation of cultural media by mixing with the powder for DMEM. To evaluate the antioxidant property of water prepared in non-contact method, the number of photons emitted per second was measured every day for 3 minutes at a time after adding 0.5 milliliter of 10 milli third buty hydroperoxide to 1 milliliter of deionized water and non-contact rated deionized water, respectively. For the duration of 4 days, TBHP as an organic peroxide is a strong oxidant and forms radical species of the form rho during the decomposition of OO bonds. It's more stable to compare with H2O2 and widely used as a source of free radical in cellular activity as well as an inducer of oxidative stress. Başlık 2.2 Preparation of Culture Media. Başlık 2.2.1 Culture media prepared by mixing with ceramic powder to avoid any possible contamination. The Kelby ceramic powder was heat treated using an electrical muffled furnace for an hour at 800 degrees centigrade degrees and then autoclaved just before mixing with Dalbicos modified Eagles medium DMEM with 10% of heat inactivated fetal bovine serum as a supplementation and 1% penicillin streptomycin was prepared by mixing the powder form DMEM with deionized water. The liquid form DMEM was mixed with 1% in weight concentration of the powder and agitated for 5 minutes and then left for 2 days in a refrigerator maintained at 4 degrees. After 2 days, the suspended rail was filtered with Wattman filter paper, grade 5, and the weight of the filtered paper was measured in order to estimate the total dissolved powder in the media. The filtered DMEM was diluted with a new fresh DMEM to certain concentrations, e.g. 25, 50, 100, 200, and 400 micrograms per milliliter prior to preparing the culturing media. The concentration of the powder in the filtered DMEM was calculated to be about 700 ppm, 750 ppm. The final concentration of powder in the diluted media was estimated to be lower than 1 ppm. For the natural killer cell and splenocyte cell, RPMI 1650 medium use was used instead of the MEM. Supplementary drawings as figure 1 to 3 in supplementary material available online at this link show the overall diagram of media preparation and cell viability measurements. Header 222. Culture media with the water prepared in the non-contact method. Another set of culture media was prepared using a structured water prepared in a non-contact manner. A centrifuge conical tube containing 50 milliliter of deionized water was inserted into the ceramic powder for 48 hours or other specified durations at room temperature. Deionized water treated in this way was used to prepare the culture media by mixing with the DMEM powder. Supplementary drawings as figure shows the overall diagram of media preparation and cell viability measurements for the non-contact method. Header 2.3 Evaluation of bioactivities of cells. Header 2.3.1 Viability of macrophage cell. Murine row 264.7 was obtained from the Korean Cell Line Bank, Korea, to evaluate the viability of the macrophage cell. It was seeded in a 96 well plate at a density of 1 times 10 to the 5 cells per milliliter in a final volume of 100 microliters and allowed to adhere for 3 hours. It was then pretreated for 1 hour with 10 microgram per milliliter lipopolysaccharides for the stimulation before applying the prepared media. After 1 hour, prepared media with increasing concentrations of dissolved Calbi ceramic powder was added to each well before incubation. Incubation was performed for 24 hours or 48 hours at 37 centigrade degrees in humidified 5% carbon dioxide atmosphere. After the incubation, media in the wells were suctioned and 100 microliter of fresh media with 10 microliter of CCK8 solution were added per well and then incubated at 37 centigrade degrees for 2 hours. A cell counting kit CCK8 Dojindo, Japan was used after 2 hours of treatment for the determination of cell viability using enzyme-linked immunosorbent assay, ELISA, microplate reader. Taken Switzerland for absorbance at 450 nanometers as a controlled the viability of the cell cultured in DMEM without LPS stimulation was measured. The viability of the cell with LPS stimulation was measured also as a positive control. The viability of the treated cells was expressed as the percentage of control cells. A schematic diagram of the media preparation and cell viability evaluation is described in supplementary drawing as figure 1. Header 2.3.2 Phagocytic activity. The phagocytic activity of a macrophage cell was evaluated by measuring the uptake of neural red dye. Cells were cultured at 37 centigrade degrees with the prepared media in humidified 5% carbon dioxide for 24 hours and then 100 microliter of 0.075% aseptic natural red solution was added and cultured again for 1 hour. Following the end of culture, the plate was washed 3 times with phosphate buffered saline and a mixture of 100% ethanol and 99.9 acetic acid was added to the 150 microliter of cell lysate. The mixture was mixed fully and absorbance at 550 nanometer was measured using an ELISA reader. The schematic diagram of the media preparation and evaluation of the phagocytic activity is shown in the supplementary drawing as figure 2. Header 2.3.3 Natural killer cell activity. For the evaluation of the natural killer cell activity and viability of splenocyte mice were sacrificed and their spleens were collected aseptically. To prepare the media, 0.1 gram of Calbi ceramic powder was mixed with 10 milliliter of Roosevelt Park Memorial Institute 1640 medium containing 10% fetal bovine serum and 1% penicillin, streptomycin, and stood for 24 hours at 4 degrees. The suspended layer in differing amounts were used for the preparation of culture media. For example, 25, 50 to 100 and 200 microgram per milliliter by mixing with a new fresh RPMI 1640 medium. Target cell was seeded in a 96 well plate at a density of 5 times 10 to the 3 cells per milliliter for 3 hours. An effector cell isolated splenocyte and target cells were mixed at effector target ratio of 10 to 1 and incubated with the appropriate media for 24 hours. NAK, natural killer, native killer. Natural killer? Yes, natural killer. Cell activity was assessed after treatment with CCK8 assay solution for 2 hours. The natural killer cell activity was calculated as follows based on the optical density measured by a laser reader at 450 nanometers. Supplementary drawing as figure 3 shows the overall cell culture process and natural killer cell activity measurement. Header 234. Splenocyte and cytokine expression. Mice spleens were collected aseptically to prepare spleen cell suspension. The evaluation of splenocyte viability was carried out according to the CCK8 method. Splenocytes were seeded in a 96 well plate at 1 times 10 to the 5 cells per milliliter for 1 hour. The prepared media with different amounts of the suspended layer 0, 25, 50, 100, 22, 100, 200 microgram per milliliter from the RPMI 1640 mixed with the 1% of Calbi powder stood for 48 hours, were added to the cells and incubated for 24 and 48 hours. CCK8 reagents was added to the cell suspension and the optical density was measured 2 hours later at 450 nanometer using a microplate reader. Changes in cytokine expression of murine splenocytes were measured also. Splenocytes were seeded in a 6 well plate at 5 times 10 to the 5 cells per milliliter for 3 hours and then incubated for 24 hours with the prepared culture media. Total RNA was extracted using trizol reagents according to the manufacturer's instructions. 1 microgram of RNA was used to acquire the complementary DNA, cDNA, using the protocol provided by MULV reverse transcriptase for reverse transcription polymerase chain reaction. Specific primers were used to amplify different gens. PCR products were then separated by electrophoresis using 1.5% agarose stained with ethidium bromide followed by a ultraviolet transsimulation. A PCR analysis was done 3 times and the densitometry analysis was carried out using the Lane ID software, 1D software. Beta-actin, interleukin, and interferon gamma expressions were analyzed. Header 235, breast cancer cell. The viability of the human breast cancer cell, MCF7, was measured in the same way using both kinds of culture media. The suspended layer was taken in different amounts from the premixed DMEM with 1% of Calbi ceramic powder after 48 hours of storage in a refrigerator maintained at 4 degrees. Additionally, to see the effect of water preparation process, DMEM mixed with the 1% of Calbi powder was agitated for 3 minutes and then centrifuged at 3000 RPM for 5 minutes. Different amounts of these supernatants were collected and used for the preparation of culture media. Also, the DMEM culture medium was which was prepared using only the water that was treated for 48 hours in a non-contact method, was used for the evaluation of cell viability. Header 2 for statistical analysis. All experiments were performed for the specified number of n indicated in the figure captions. From n is equal to 4 to n is equal to 7, and results are presented as mean plus minus ST, standard deviation maybe. These cellular activities in intact cultures were taken for 100% of the culture. Statistical significance was determined using one-way analysis of variance, followed by Duncan's multiple range test. A value of p is smaller than 0.05 was considered statistically significant. Header 3, results. Header 3.1, antioxidant property of water. Header 3.1.1, water mixed with the powder. The Kelby powder showed a good hydrophilic property, and colloid formation was observed when mixed with water. For the bean sprout stems mixed with the water prepared in a contact method, the number of photons emitted are not much different among the three specimens, measured 16 hours later as shown in figure 3a. However, the number of photon emissions from the deionized water mixed with silica powder, red line, increased at 6 days, followed by deionized water, only blue line, as shown in figure 3b. After 7 days, it was almost the same as shown in figure 3c. In comparison, the deionized water mixed with the Kelby ceramic powder, green line, clearly showed a lower photon emission even after 7 days, as shown in figure 3b and 3c. Table 1 shows the average number of ultra-weak photon emission per 1 second measured for 8 minutes from the bean sprout stems mixed with different kinds of water prepared in the contact methods. In the deionized water and deionized water mixed with the silica powder shown as the figure 4a, 4b, and 4c, the color of the bean sprout stems turned dark brown and brown. However, the color of the bean sprout stems stored in the deionized water mixed with the Kelby ceramic powder did not change significantly and was light brown after 7 days, as shown in the figure 4c. These results indicate that bean sprout stems in the deionized water and deionized water mixed with the silica powder oxidized faster than those stored in the deionized water mixed with the Kelby powder. In order to indicate the change of the color quantitatively, an arbitrary unit from 1 with no change to 5 dark brown severely changed has been adopted. Figure 5 shows the variation of color with the incubation time, 16 hours, 6 days, and 7 days for the bean sprouts shown in figure 4. Until 6 days of incubation, there was no difference in color between the ionized water and in the ionized water mixed with Kelby ceramic powder. Next day, on the 7th day of the incubation, the color of the bean sprouts in the deionized water mixed with the Kelby powder hardly changed, but that of the bean sprouts in the deionized water began to change. Figure 3, measurement of ultraviolet photon emission with a photomultiplier tube. The blue line, red line, and the green line indicate the number of photons from the ionized water only. The ionized water mixed with the silica powder and the ionized water mixed with the Kelby powder, respectively. The number of photons per 1 second was measured from 8 minutes. The vertical axis depicts the number of photons, and the horizontal axis represents seconds elapsed. The color of Beans protein deionized water mixed with the silica powder rapidly changed continuously from white to dark brown from the beginning all the day to the last seventh day. Header 312. Water treated in the non-contact method. Experimental measurements show that the photon emission from the deionized water increased on the second day of measurements. On days 3 and 4, the number of photon emissions was lower than those from day 2. However, they were still higher than that of day 1, as shown in figure 6a. In comparison, the number of photon emissions from the deionized water treated in a non-contacted manner showed a dramatic decrease on day 4, reaching 1/4 of its initial level after 93 hours of exposure, as shown in figure 6b. Figure 7 is redrawn from the data shown in figure 6 for direct comparison to the control data. After 16 hours of the non-contact treatment, the treated water already shows a lower number of photon emissions compared to the control as shown in figure 7a. This difference increased after 93 hours of exposure, and its level was about 1/4 of the control. This indicates that the antioxidant property of the water treated in a non-contact manner increased as exposed time increased. Figure 4. Observation of the color change of bean sprout stems. Deionized water mixed with the silica powder, deionized water with the calcium ceramic powder, respectively. Effects of cultural media on the cell viability. Header 321. Culturing media prepared by the mixing with powder. The viability of raw 264.8 macrophage cell increased in a dose-dependent manner in the amount of the added suspended layer in the as shown in figure 8. Compared to the control, it increased 1.5-fold in a media of 2 microgram per milliliter concentration. There was no significance in the viability of cells, regardless of incubation time length. Note that all of the samples incubated with the prepared culture media after LPS stimulation showed a higher viability compared to the samples treated with LPS stimulation only or incubated without LPS stimulation. The prepared culture media increased the phagostic activity also, as shown in figure 9. The activity almost doubled for the concentration of 5 microgram per milliliter, and almost the same level of activity was maintained up to this 80 microgram per milliliter. Unlike the viability, the dose-dependent manner was not observed. For reference, the activity of raw 264.5 macrophage cells stimulated with the LPS only increased about 1.5-fold compared to the control. A measurement of natural killer cell activity showed that the prepared media has a positive effect in a dose-dependent manner, as shown in figure 10, resulting in a 3-fold increase for the medium mixed with the 200 microgram per milliliter of the suspended layer. The incubation mouse splenocytes with the prepared media showed a significant dose-dependent effect on the viability, as shown in figure 11, although it's not as dramatic as in the case of phagocytic activity. The reverse incubation. Figure 5. Variation of color of the bean sprout stems with incubation time. Arbitrary units in 5-point scale in adopted to indicate the color change. That's from AU1 white and no change in color to AU5 dark brown and severely changed. Transaction polymerase change reaction shows that both interleukin and interferon gamma expressions were up regulated dose dependently by incubating with the prepared media as shown in figure 12. This result confirms the increased activity of the splenocyte cells shown in figure 11. Header 322. Culturing media prepared with water treated in the non-contact method. For the culture medium prepared by mixing the DMDMEN powder with only the ionized water treated in a non-contact manner, the viability of macrophage cell was almost doubled and tripled dependent on the length of the incubation time as shown in figure 13. Since the macrophage cell is sensitive to stimulation from outside as an immune cell, other kinds of normal cells besides macrophage cell, including Rhodes, norvegis, mouse meioblast, and murine adipocyte, were incubated in the same way to confirm whether this same tendency is observed or not. Although there were some differences in the level of viability, all of them showed that the same tendency, that's increased cell viability as shown in figure 13. The effect of incubation time was not so large compared to that of the macrophage cell. The unexposed deionized water was used for the preparation of control for all kinds of cells. To observe the effect of difference in exposure time on the viability of macrophage cell, culture media was prepared using deionized water which was treated in a non-contact manner for 6, 12, 24 and 48 hours respectively. All of them showed an almost threefold increase in viability with a slight difference among them as shown in figure 14. This means that even a short period of exposure time shorter than 6 hours may be enough to increase the viability of macrophage cell. Header 323. Effect of cultural media on a breast cancer cell. We performed the same experiment to observe its effect on the viability of an abnormal cell. A breast cancer cell MCFC7 was incubated in the same manner and the viability was suppressed in a concentration-dependent manner as shown in figure 15. It was inhibited to a level below 80% compared to the control for a concentration of 200 microgram per milliliter. The same trend was observed in the media prepared with a water treated in non-contact manner for 48 hours as indicated by the bar of EQ in figure 15. To see whether there is any influence on the cell viability of preparation process, the Calbi powder cell, the ceramic powder mixed with the MDM and centrifuged for 5 minutes right after mixing the powder rather than waiting for 48 hours of standing time. The supernatant after the centrifuge was used for the preparation of cultural media in a different amount. Results shown in the figure 16 indicate that the effect is slightly lower for the same concentration, 100 and 200 microgram per milliliter each, compared to those of data shown in figure 15. At the concentration of 400 microgram per milliliter, the viability was suppressed almost the 70 of the control. This result implies that 48 hours of standing time may not be necessary to see the effect of the cultural media prepared in a contact methods. Discussion. Header 4. Discussions. There has been an argument regarding to psychochemical properties of interfacial water and the possibility that it may influence biological activities depending on the hydrophobic and hydrophilic properties or hydrophilic properties of the cell membrane and other organic components of cells. It was claimed that water in contact with a hydrophobic surface will have a lower density and better absorption of various surfactants and proteins from water. This approach is based on the variation of the density of the interfacial water. This approach is based only on variation of density of interfacial water. It has been reported that the increase of ultraviolet It has been reported that the increase of ultraviolet photon emissions is associated with an oxidant reaction. The number of photon emissions is reduced when an antioxidant component such as catechin is added to the water. Catechin is added to water while it increases under an antioxidant atmosphere. Therefore, the number of photon emissions is expected to decrease for water with an antioxidant property. The number of photon emissions is expected to decrease for water with an antioxidant property. In this study, it was clearly demonstrated that the structured water prepared through a mixture with the hydrophilic ceramic powder exhibits an antioxidant property. Antioxidants inhibit the oxidation of lipids, proteins, and other molecular components in cell. And the generation of an ultraviolet photon emission is also associated with the oxidation of proteins and amino acids in bean sprout stems. This indicates not only that there is a variation of the density of water, but also that an electric property can be a critical factor in the role of water. Actually, it has been found that an electric potential as high as minus 200 millivolts exists across the boundary between the exclusion zone and outside the region exclusion zone negative, in addition to the difference identity. Interestingly, the water treated in the non-contact manner also demonstrates an antioxidant property as shown in figures 6 and 7. The level of antioxidant property increases with the longer exposure time. The energy source for the formation of structured water in the non-contact manner is possibly the infrared rays radiating from the ceramic powder. This technique is quite important in the analysis of experimental results, since only this structure of water was modified without any addition of material components to the water. The modification of water structure seems to be a sort of continuous and gradual phenomenon. According to the experimental results of ultraviolet photon emission, which was carried out for 93 hours in the non-contact manner, the water can be structured for quite a long time continuously through exposure to the powder for a long time. However, the effects of exposure time on the cell viability show that less than 6 hours is enough to obtain the increased viability of raw 264.7 cell. This implies that it may not be necessary to make a highly structured water by treating the water for a longer time for biological purposes. In other words, cells are very sensitive to structure of water, which is supported by the fact that even a very small amount of premixed structured water, 0.5%, is effective in increasing the phagocytic activity. Additionally, the structured water is stable enough to show these effects even when mixed with a new fresh culture medium and shaken well. Only a small amount of structured water is necessary to increase bioactivities of cells, as shown in figure 8. The culturing media mixed with Calvi powder or prepared with the antioxidants structured water exhibit an increase in the viability of normal cells, recognizing that the same tendency was observed for both kinds of media prepared either by mixing with the Calvi powder by mixing with the deionized water treated in the non-contact manner. The experimental results reported here must be interpreted as it relates to the antioxidant property of water generated by the modification of water structure rather than the material components. It's expected that a reactive oxygen species scavenging effect may occur through this media as other nutritional antioxidants. As a result, the electron transport change in the mitochondrion membrane may be activated, resulting that increase the viability for normal cells. Considering the principle of the CCK8SA kit, this explanation is more convincing since the formazan dye is formed as tetrosodium salt in the CCK8 solution, which is reduced by the dehydrogenase in the electron transport change. Another possibility is in the increased proton concentration. When the electrical potential is generated between the exclusion zone and outside of it, a large amount of protons is generated in the outside region. This means that the supply of proton can be enhanced whether water is structured. Considering that the fact that the mitochondrial membrane potential is composed of potentials from ion and proton gradients, the increased amount of proton may contribute to the increased gamma psi, increased psi. Actually, it was reported that CM was increased up to 1.36 fold for rho, 264.8, and HD11 cells after exposing them to the Kelby powder in the non-contact manner for 48 or 72 hours. The increased PCM may contribute to the increased viability and increased ATP production. However, this was not observed for these cells. On the other hand, the same culturing media show the opposite trend for malignant cancer cells. This is quite an interesting phenomenon since the same stimulator results in the opposite reactions depend on the cells normal or malignant. At this time, the mechanism is unclear, but the increased CM may provide clues for further research. According to the previous report, the CM was observed to increase also for the HeLa cell by more than 1.36 folds, while the intracellular ATP level decreased after 72 hours of exposure. It's well known that cancer cells have a more hyperpolarized Psi than normal cells and can be larger than 50% than normal cells. It has been reported that 10% value alternations from its optimum in CM can cause serious variability on the ATP synthesis by as large as 90% degrees. Thus, the hyperpolarity in CM of a cancer cell may be related to the malfunction of electron transport chain. That's oxidative phosphorylation. Warburg reported that cancer cells metabolism occurs through aerobic glycolysis unlike normal cells, which rely on aerobic respiration. The increase in Psi and decrease in ATP production in the HeLa cell cultured in the non-contact manner with the Kelby powder imply that the variation in PCM is one of the causes for decreased viability of cancer cell. MCF7. Definitely experiments on the effect of structured work on the potential PCM is required for a detailed mechanism. It has been claimed that continued oxidative stress can lead to chronic inflammation, which in turn could mediate most chronic disease, including cancer. In the report, the antioxidant property of structured water, increased viability, and stokin expression and suppression of cancer cell were described. More detailed anti-inflammatory effects of Kelby powder were reported elsewhere. It's presumed that all of these studies, antioxidant, anti-inflammation, anti-cancer effects of Kelby ceramic powder, are strongly correlated to each other. Additionally, it was observed that DNA exposed to Kelby was protected from the damage caused by free radicals. These results suggest that a future study on the effects of Kelby should focus on its overall holistic influence on a living body rather than focusing on molecular level cell cycle regulatory system. Again, These results strongly suggest that water plays a critical role in cellular metabolism more than previously understood. Water is not merely a simple liquid, but instead plays an important role in cellular and molecular mechanisms by stimulating the enhanced bioactivities of normal cells, while suppressing those of malignant cancer cells. All of these results strongly point to the positive role of structured water in cellular bioactivities. Conclusion. Header 5 conclusion. The experimental results reported here imply that both the materialistic nutritional components of culture media and water structure can be critical factors in the analysis of bioactive activities of cells. Structured water, either prepared through mixture with a hydrophilic ceramic powder or treated in non-contacted manner, exhibited antioxidant property. The increase in cellular bioactivities is strongly related to the antioxidant property of structured water. Structured water appears to exhibit the same properties as an antioxidant. Since no antioxidant components were added in the water treated in the non-contacted method, these properties are solely related to the structure of water. These experimental results strongly suggest that water can play an essential role in bioactivities of cells, depending on its antioxidant effects based on its structure. Water itself can be an active constituent in cell biology, as are many other cellular molecules. Disclosure.